So today I'm going to talk about some work that we've done on blue waters to refine the conformational ensembles of very flexible proteins. So flexibility means different things to different people. So I want to place myself along the spectrum of protein heterogeneity. So at one end, there are these very rigid structures, which are primarily governed by their side chain fluctuations, and there isn't a whole lot of backbone rearrangement. And then on the other end, you have something like intrinsically disordered proteins, where it's all backbone heterogeneity. And so you can, in many cases, treat those with coarse-grained models or even polymer theory. And so all of these different, classes of proteins have their own set of challenges for refining their ensembles. But I think that some of the toughest ones to study are the ones that sit somewhere in the middle. So these are not so flexible that you can study them with coarse-grained models, but they're also not so rigid that you could look at them with something like X-ray crystallography and study just a few low-energy states. So a big part of the challenge is that it's hard to go from experimental data to high-resolution estimates of these ensembles. And that's because the experimental data tend to come in sort of two forms. They're either ensemble average quantities, which are going to tell you about average properties of your system, but not about specific conformations that make up the ensemble. And then there are experimental techniques that yield distributional data, like DEAR and FRET, and these are experimental techniques that give you specifically distance distributions between pairs of labeled amino acids. So this is great because it reports on heterogeneity, but oftentimes these data are sparse over the atomic coordinates. So for DEAR and FRET, you can really only get, on average, about order 10 distributions for a particular system, whereas you would need, thousands to fully characterize all the atomic degrees of freedom of something like this. Okay, so a lot of great work has come out on using ensemble average quantities to back out estimates, high-resolution estimates of the ensembles. So these are things from Markov models all the way to Bayesian methods for backing out the ensembles, and these work quite well. What people haven't done as much of is try to leverage these sparse distributional data to back out an estimate, a high-resolution estimate of the ensemble. So what we've done is we've developed an iterative method for using these sparse distributional data to refine these kinds of heterogeneous ensembles. And we've called this method bias resampling ensemble refinement, or BRer. So how does this work? Here on the left is a general idea. We start with an initial estimate of the conformational ensemble. So this could be like an NMR ensemble maybe, or even a single crystal structure, but some experimentally informed estimate of the ensemble. And then we draw some number of conformations from the ensemble with replacement, so n conformations with replacement. Now, suppose that we have some experimental distribution that we want to use to inform our model. So here I'm just showing a sort of schematic DEAR distribution. We'll end up working with DEAR, but of course you could use any distributional data. Okay, so you have this experimental distribution, and we're going to do stochastic resampling from the experimental distribution. So essentially I'm going to do n probability-weighted draws from the experimental distribution. I'm going to pull essentially n distances, if this is deer, and I'm going to distribute them. So each distance is going to go to a particular ensemble member that I've drawn up here. And I'm going to use biased molecular dynamics simulation to drive each of the conformations to its particular target. So I'm refining each conformation against one distance value that I've drawn. So then once the ensemble has relaxed at the appropriate targets, then we update our estimate of the conformational ensemble. Essentially, we dump our new conformations into this conformational bag, and then we repeat this iterative process until we are satisfied that the simulation distribution agrees well with the experimental distribution. So there are sort of two conceptual pieces to this method that I want to talk about. One is this idea of stochastic resampling. So what I'm showing up here is just this process of drawing a set of targets and how that leads to convergence on the experimental distribution. So here I've eliminated any MD. What I've done is I've replaced it with a Gaussian stub. So basically what I say is whenever I draw a target, my model is essentially going to sample a fairly tight Gaussian centered around the target that I've drawn. Okay, and so you can see that even when you remove the biased MD, you can converge on the experimental distribution relatively rapidly with about 500 total targets. Okay, so that's one piece of this method, and then the other piece, of course, is the biased MD. So here, forget about the stochastic resampling. We're just picking one target, and we're trying, and it's one iteration, so we're trying to drive one ensemble member to a particular target. So in this case, we're going from a distance of 2.5 nanometers to about 6 nanometers. And so the way that we do this is by adding a linear biasing potential to the Hamiltonian, which might be a little bit hard to see here. But we use a method out of Greg Voth's lab, actually, for training an optimal coupling constant. That's this alpha here, which might be a little hard to read. So this is actually something that the speaker last night talked briefly about, how you can use machine learning to essentially improve your MD simulations, and that's what we're doing here. We're selecting, well, we're using gradient descent to learn an optimal value of this coupling constant so that we inject just enough energy into the system to get our protein to converge on the target, but not so much energy that we destroy the secondary structure, right? Okay, so moving on to actually testing this method. We tested this on the SNARE protein syntaxin 1A. I don't want to go deep into the biology here, but just a quick overview. SNAREs are protein complexes that drive neuronal vesicle fusion. And one of the proteins that participates in formation of the SNARE is this syntaxin 1A. And this protein has a single transmembrane domain and then a soluble domain that interacts with the 3 other proteins that are involved in formation of this complex. So the soluble domain has an open and closed state, and we know that syntaxin is in its open state when it binds to these other SNARE proteins. But that's really about all that we know about the conformational equilibrium of this system. So we have some collaborators at UVA who are really interested in the conformational equilibrium and who performed some DEAR experiments to try to investigate that. But they needed a way to integrate their sparse data into a high-resolution estimate of their conformational ensemble. So we decided to use BRer to integrate the data. So here's what we did. We refined the soluble syntaxin conformational ensemble against 3 DEAR distributions, which are shown in the black lines in this plot. And the BRer refined ensemble, which is the pink distribution, shows very good agreement with the experimental data. We also compared the BRer refinement method to 2 other existing methods for doing this sort of refinement. One is based on metadynamics, that's this EB meta D in green. And then another is a restrained ensemble, which is shown in blue. Now, these other methods are really great at reproducing somewhat less heterogeneous distributions. So really, they've been designed to handle sampling of smaller fluctuations like what you might see just from the rotameric states of the DEAR probes. So they're not explicitly designed to handle large backbone movement, which we explicitly designed BRer to do that. So at any rate, excuse me, the BRer refined ensemble shows much better agreement with the experimental distributions, and we quantified that over here on the right using an information theoretic metric called Jensen-Shannon divergence for people who think about information theory. But it's basically a quantitative measure of how well 2 probability distributions agree with one another. Okay, so technically this all looks very nice, but what did we actually learn about the syntaxin conformational ensemble? So to explain that, I need to give you a little bit more detail about the soluble domain. It has 2 motifs. One is this HABC domain shown in magenta, and then this H3 domain in green, and these are connected by a linker region. Okay. So the sort of canonical understanding of the syntaxin open state is that this linker region unwinds, and the H3 domain essentially retains its secondary structure and just peels away, dissociates from the HABC. So the canonical open state is shown here in tan. The BRer refined simulations actually revealed an additional, previously unreported, sort of partially open state where the linker region actually retains its structure, but then the H3 domain is fairly disordered. It loses its secondary structure, right? So both of these structures are consistent with the DEAR data. So this leads us to a potentially new hypothesis, which is that the interaction of syntaxin with the SNARE complex could actually be the result of a conformational selection process where you have a much more heterogeneous open state than was previously thought, and it's these conformations that are selected out, the ones where H3 is ordered. These are the ones that actually participate in formation of the SNARE. Now, one caveat, I can't tell you today that we're 100% sure that this partially open state really exists. We need to do more experimental measurements to confirm that. But I think that this result still shows the importance of developing these kinds of rigorous methods for incorporating, for incorporating these kinds of measurements and for refining these flexible ensembles. So with that, I know that my time is up, so I want to thank everybody in my lab, especially our postdoc Eric Ergang, who's been developing a Python interface for GROMACS called GMX API. And actually, my advisor Peter gave a talk on GMX API last year, so if you're interested in interfacing with GROMACS in a much easier way using Python, you can check that out. That's probably floating around the internet somewhere. And then our experimental collaborators and funding, and then of course Blue Waters. So, whoops. Oh, come on. Okay, so sorry that this slide isn't very glamorous, but each of these ensembles were 5 microseconds worth of all atom simulation data, and so pettascale systems like Blue Waters are of course very important for this research. I also want to especially thank Blue Waters for inviting me back as a returning 2017 Blue Waters graduate fellow. So the experience as a fellow was awesome, and I can't thank you all enough, especially the people at NCSA for their support in getting my codes up and running. I do not think that I would have published either of these papers if I hadn't had both the resources and the support from the staff.